You know, we do miss Vermont. I first went to Vermont with John and Marna and Chuck and the whole crew 2 years ago, totally wet behind my ears, just about to start as the scientific director for this foundation. And an overwhelming memory from that time was sitting down by the fireplace with some of the younger patients who were there and talking really deep into the night, 2, 3 in the morning, about their stories. And at the time, and even still today, most of you who have dealt with this cancer know that we're still dealing with the old-fashioned triad of treatment options that have been around for cancer for a very long time. If you're fortunate enough that your tumor has not spread badly, even if it's a large liver tumor, you're initially going to undoubtedly seek out surgery, maybe in a very extreme case, a liver transplant, but more likely resection. You may have some radiation as part of your procedure. And depending on your situation, especially if the cancer has evidence of having spread outside the liver, you'll have some form of chemotherapy. You know, if you have a couple of big lung mets, maybe the surgeon will take them out, but you're certainly not going to be cured of that kind of disease by what we have today. There's no standard of care. There are few cases with proven long-term benefit from chemotherapy or any other therapy other than surgery. Not a great picture. And yet we're here talking about maybe there's a possibility to use the word cure. And some of this is really because there's a modern world of cancer therapy, which is emerging and maturing and becoming really the standard of how we try to do research in cancer. And the terms that are going to be used tonight and you'll hear over and over are targeted therapy on the one hand and immunotherapy on the other. Today, I would say that the vast majority of our scientific roadmap for how we're going to try to get to curative therapies for this cancer within your lifetimes surround those two concepts. So I'm going to talk about them, and I'll try to stay away from a lot of pure lab or science speak and try to, as Marna always asks me to do, speak English. And if I fail at that, I hope the questions will come fast and furious at the end. But let's talk about what targeted therapy is. A lot of targeted therapy recognizes that cancer is a genetic disease and that mutations in certain proteins create altered proteins that can drive the cancer. Other mutations occur, but have less of an effect, and we sometimes call those passengers. So this is the driver as in the driver of a bus. It's what makes that cancer continue to grow out of control and create the havoc that we're ultimately wanting to stop. So one of the key areas is to understand the kinds of growth signals that are involved in driving cancers, and specifically fibrolamellar, of course, and what are the biochemical pathways that maybe one can hope to interfere with in order to put up a stop sign to that growth, or maybe even more than a stop sign to get the cancer to commit suicide, if you will, to die. Another part of targeted therapy has been understanding more about the metabolism of cancer in the context of how it differs from normal tissue and whether that's something that can be leveraged for better therapy. Another area that's somewhat related to that is trying to choke off the blood supply to cancer, because cancers, of course, need blood supply, particularly when they're rapidly growing. So you can sort of take almost a humorous view of it, do not feed the wildlife, but seriously, this is a new part of the armamentarium, which is different than old-fashioned chemotherapy, which is essentially throwing toxins at cells and hoping that they're just that little bit more toxic to the rapidly growing cancer than to your normal tissue. An extreme version, and one that I'm actually quite excited about in cancer, what you might call molecular smart bombs. And that's really finding better and better ways to use what might in fact be very old-fashioned kinds of toxins, but to direct them extremely precisely only to the cancer cells so that you don't get the side effects that you see today. And we're moving to a generation where one of the tools for delivering those are antibodies. There are other ways to deliver them, which can recognize very precisely some aspects of the cancer cell surface that are different from a normal cell. We're also in a position where the nature of those payloads, the toxic compounds, and the way they're linked to the delivery systems are getting better and better. So these are the kinds of areas we'd like to develop rather than the more traditional kind of chemotherapy. And there's nothing new or special about this. This is where the whole field is going. The second major modality, which has really come into its own in the last few years, although as John said, it's something that people have been dreaming about for decades, is immunotherapy. And really the goal here is to enable the patient's own immune system to do something that's been evolved to do and programmed to do for millennia, or for as long as we've had life on this planet of a multicellular variety, which is to find renegade or rogue cells and eliminate them before they damage the whole organism. Cancer's been with us on the planet for a long time, and the immune system's pretty good at smelling it out and stopping it from growing. Unfortunately, cancers also evolve in many cases to bypass the surveillance of the immune system. Cancers can put up a kind of defensive shield, taking advantage of what are technically known as immune checkpoints. And there's been a new generation that many of you have heard about of what are called immune checkpoint inhibitor drugs. I'll probably just call them checkpoint drugs at most of the talk right now. And I think of those as arrows that can pierce that shield, or if you want to get a little more fanciful, sometimes those of you who are still Star Trek fans think about a cloaking device which protects the cancer, and these drugs are a decloaking device so that you can see it if you're an immune cell and go after and try to kill it. A second class of therapy that is related is to, in some cases, see if one can actually program a patient's immune cells to do a better job of attacking the cancer. And there are, in fact, a class of cells that we call T cell killers or killer T cells that are exquisitely specific in targeting certain classes of cells that the body needs to get rid of. They might be virus-infected cells, they might be cancer cells. Can we make them do a better job? Can we use genetic engineering or some other aspects of biology to increase the number of these cells, increase their specificity, and protect them from being blocked by the shields that the cancers can put up? So these are areas we really want to think about hard. Another area, and I won't dwell on it today, although we'll talk about this in the context of the biobank, that is crucial is not only to develop the therapies, but really to have the infrastructure that allows one to do the research biology and the drug discovery in a way that will get us to the goals for fibrolamellar itself. As is true for many rare cancers, we don't have some of the same laboratory resources for fibrolamellar that people would have for colon cancer or lung cancer, where different kinds of laboratory models have been developed for decades. So we need to be able to do a better job of growing human fibrolamellar cancer cells in laboratory culture. That starts with having the actual cells from patient biopsies available, and that's where the biobank comes in. We also, as the field has learned how to do for some decades, sometimes grow those tumor cells as tumors in mice that don't have good immune systems. And so these are sometimes called PDX models. You may have heard that term. Those two ways of working with the actual human cancer cells are crucial to making progress. Another thing that we'll do is use animals not to grow human cells, but to mimic them. One can put into the mice, for example, the same kind of driver gene that causes fibrolamellar or causes any other cancer, and that cancer driver gene can, in fact, generate a tumor that in many cases mimics the human cancer pretty well and allows one to do a variety of experiments that in some cases you can't even do with the human tumor cells, like look at a cancer in an animal that has its own intact immune system. So in fact, that tool was critical in work that was, I think many of you know, really sort of seminal in the field and allowed the real big intellectual breakthrough and practical breakthrough that's fueled so much of the research roadmap in fibrolamellar for the past 6 years. The Simon Laboratory at Rockefeller University, at the time funded by the foundation, made an important discovery that in a set of fibrolamellar patients, every one in that individual's tumor had a very specific kind of mutation, and one that's found in other cancers, but a little unusual relative to many cancers. It turned out to be a loss of DNA, a deletion, as we would call it technically, that brought two genes that are otherwise separate and unrelated very much closer together in such a way that it actually created a hybrid or chimeric gene, or fused gene. These terms are all synonyms. Shown here in this little picture with yellow for the part that comes from a gene that codes for a protein called protein kinase A, and another one that codes for a heat shock protein with the funny name DNAJ. But we'll just call that the chimera tonight. And although, technically you really should probably have a centaur, not a three-headed or a two-headed beast with a third organism's tail, because we've only got two components. The chimera, however you name it and however you think about it, opens the door to targeted therapy of fibrolamellar cancer, because in the pure form of the disease, it looks like maybe as many as 99% of patients share exactly this driver. And that's something that's really unusual in cancers. And it gives us the opportunity to really focus on understanding the signaling and the pathways that we might want to go after. And if we can generate a cure, it's one that could apply to a very large fraction of the patient population. An important discovery over the last couple of years, and I'm going to try to give you a couple of highlights that I think are important towards our progress, was from the laboratory of John Scott in Seattle, Washington, at the University of Washington. Other labs are working on similar things, and there's been related contribution recently from a terrific lab in San Diego. But the understanding is that one has to think not only about this driver protein itself, which is shown here in a little green circle in a cartoon, where the pKa part, the main part with sort of the big activity, the protein is in blue, and this little extra head from the other gene is shown in yellow. So that protein is actually part of a bigger complex, and it interacts with proteins that are normally there as part of growth control and kind of a signal exchange or a relay for information in the cell that, among many other activities, is really pivotal in whether the cell is going to actively grow or not. And if something goes wrong here, there's the potential the cell will grow when it's not supposed to. One of the discoveries that the Scott lab has made is that because this is a chimeric protein and has this little extra domain, it qualitatively behaves in a different way from the normal pKa, which is a very well-known enzyme. In fact, John worked for some years with the discoverer of that enzyme and holds a chair at the University of Washington named after his mentor, who was a Nobel laureate. But we now know that pKa itself, even if it's made at higher levels than normal, does not transform a cell and create fibrolamellar cancer in that mouse model we just talked about. The fusion protein, the chimera, qualitatively is different. It has a much more potent activity in causing cancer. Why is that? One possibility is that it brings another protein into the complex that's not normally there. And that may change the interaction with a bunch of other proteins, and don't worry about what their names are, but these are well-known proteins involved in growth signaling. And there's something about this aberrant complex which makes these more active and less easy to turn off in giving growth signals, so that they drive the growth of liver cancer cells. One of the thoughts that John has published with his group is that it may be that actually targeting a combination of proteins in that overall complex may be more effective than going after any one. Obviously, the most ideal thing would be if you could actually knock out the chimera itself, but there are technical reasons that that may not be so easy without causing damage to the body, because the protein, the blue part here, is also really critical in other functions like how the heart works, how insulin works, and so on. So we don't know if we can go after that, but maybe we can find novel combinations that will work on these aberrant complexes. Another approach that's been taken, and this has been led by Praveen Sethupathi of Cornell University, who's been working on fibrolamellar now for about a decade, is to ask, can we just identify genes that the fibrolamellar cancer needs to survive or to grow? And this is part of a much bigger picture. We're, for example, collaborating with the Broad Institute of MIT and Harvard on looking at that question systematically. Praveen has used tools that geneticists have developed to understand what genes are just locked on in the cancer by things in the chromatin, or the controls how genes are used, that have the great name of super enhancers. So they're locked on and are turning out certain proteins in a way that you might say the cancer is addicted to. It needs those. And among the set of genes he's identified that have that property of really being in that constant on and driven state in fibrolamellar are some attractive new candidates that might be targets for therapy, some of which you might never have thought about in other contexts. So that's yet another approach that, in fact, again, one would like to take even more broadly. So just to summarize where we are with this approach to targeted therapy, again, an ideal thing would be if we could find a way to either selectively block the activity or maybe the production of the chimera. And in fact, we're embarking in some collaborative work through the Cornell group and a company, Alnylam, on one strategy that might allow us to block the production of that fusion protein. There are others that we have at least outlined on blackboards and are hoping to find people to work on over the next year or two as part of an expanding roadmap in that area. As we already said, inhibiting downstream steps that control cell growth pathways that are ultimately driven by the chimera is another possibility. If you could find targets that either already have existing drugs in development or combinations of those, perhaps with one existing drug, something still waiting to be developed and so forth. But these possibilities of combinatorial approaches based on better understanding of the biology are very attractive. And again, we said you could target genes that are locked on by super enhancers, and also to use the models that we've talked about to enable the largest possible scale of screens for targets that are necessary for survival of the cells and drugs from large sets that just really, every compound that's ever been shown to be safe in a human being, which of those might be beneficial against fibrolamellar. And in fact, that's part of our long-term collaboration with the Broad Institute. I want to turn now to immunotherapy, and I'd like to start with just what I think is a terrific graphic view of, nice if I could get the movie to play. Let's go again. I wanted to show you what happens when the right kind of T cell finds a cancer cell. Maybe I'm going to have to send you to this another time. Sorry about that. There we go. So green here is a killer T cell. The blue cell in this video is a cancer cell. The two little red dots actually show when that T cell is lining up to attack the cancer cell. It's a sign that it's found the cell. So you see those two dots get in position. That T cell is now about to start the process of zapping the tumor cell. Wouldn't it be great if we could find an army of those T cells in patients and get them to do that to the cancer cells? This is the same thing, but just with different filtering. So now that T cell appears bright golden, and you get a real idea of how intimately it's wrapped around the tumor cell as it's undergoing the very specific process of destroying it. But as we said, sometimes you can have a T cell, shown here in the bottom left panel as the blue cell with a little sort of purpley color nucleus, going after a tumor cell. And in this case, in fact, that T cell should be able to kill the cancer cell because it's got the right lock and key structure to find something that's specific about the tumor cell that is the trigger for it to want to kill it. We call that a receptor and an antigen. Don't worry about the language again. But in this particular case, something else is going on. One of these so-called immune checkpoints is intervening something that the tumor cell is matching a different surface receptor on the T cell, and it's saying, don't kill me. You're not supposed to kill me. It's overriding the normal behavior of that killer cell and putting up a shield. What the checkpoint drugs do is basically unshield the tumor cell and do that by destroying the interaction between that second signal. It gets rid of the don't kill me signal so that in fact the T cell does its job of killing the cell it should be going after. And that leads to the question, can immune T cells in fact kill fibrolamellar cancer cells? There's some reasons to think that fibrolamellar cancer cells might be what people sometimes call immunologically a little bit cold. That is, not really good candidates to be killed by the immune system. But that may not be true, and we're going to try to find out when T cells can actually kill the fibrolamellar cancer cells. And we want to ask very specifically, do FLC cells use the immune checkpoint system to evade? Do they put up the shields, and can those shields be overcome by inhibitor drugs? And just the names of some of them, which many of you may be familiar with, are shown on the slide. What we've heard so far, although nobody's done a very systematic study, there's in fact one in place at the Dana-Farber Cancer Center at the moment, but it appears that single checkpoint drugs don't do a very effective job against fibrolamellar. And that may not be so surprising, given that they're not in that kind of hot level where they have so many mutations that the immune system can find them easily. So the question is, can there be ways to enhance that? There's a clinical trial which is coming up that we're funding at MD Anderson Cancer Center that actually builds on some work that's already being done at Rush in asking the question of whether a combination of two drugs that have been used for some years in fibrolamellar, many of you may have even had those drugs yourselves if you're patients, would be a combination of 5-fluorouracil and interferon, but now asking, do those drugs potentially synergize with a checkpoint drug to kill fibrolamellar? And that's an approach that, again, mimics what's going on elsewhere in the cancer research field. Another thing which has happened in recent time is that people have begun more and more finding ways to be able to use more than one of these checkpoint drugs at the same time, which work against different shields, two different shields. And there's at least one very encouraging case report that appeared early in 2020 from a laboratory and hospital in Germany saying that one very fortunate young woman who had a very advanced case of fibrolamellar, a combination of two drugs, in fact, gave her a really strong response, and her tumor is essentially virtually gone. And I just spoke to Dr. Dottoni a couple of weeks ago, and the patient is still doing extremely well. So this dream of creating an army of anti-fibrolamellar T cells is a real one, and you can imagine kind of a Clone Wars scenario where you'd like to have not one lonely fighter with the gun that's specific for fibrolamellar, but turn that into an army. Well, how do you expand them to create an army? If you can find one, is there a way to go from one to many millions? And the answer is there are some ways. One is to use genetic engineering of T cells to create the army, sort of manufacture it, and then put it into play. But another thought is one that was brought to us by a young physician scientist from Johns Hopkins named Mark Yarchuin. He came up to John Hopper and me at a ASCO meeting in January of 2019 and said, hey, you know, maybe we could vaccinate patients, not to prevent disease because they already have it, but maybe we could create a therapeutic vaccine for fibrolamellar that would expand any T cells they have that are capable of killing fibrolamellar. And the very specific question he asked, and one which I thought was really logical, and I've actually been asking it since about the day that I joined the foundation, is can the chimera protein be a target for the immune system. Why is that an interesting idea? Because the driver of a cancer, if it becomes also the target for an immune system, gives the immune system and those who want to treat the cancer a great advantage. It's hard for the cancer to lose that driver. Usually with immune therapy, you worry, if it's just sort of an accident, the protein has a signal for the immune system to find. Maybe you can throw away that signal and still grow as a cancer. But if it's the driver gene that's being attacked, it's not so easy, and especially for a cancer like fibrolamellar that's a little more stable than some other cancers and is probably locked into the same driver much more tightly. Some cancers can generate new drivers really fast. Fibrolamellar doesn't seem to do that. So here was the idea. The green part of the protein is normal. The blue part of the protein is normal. The immune system won't react to those. The junction between the two is not there in normal cells. Maybe the immune system can pick out that junction. So what Mark Yarchman said, all right, well, let's try to vaccinate with a little piece of the protein that corresponds to that junction region and see if we can't get patients to amplify an immune response that way. And so that clinical trial actually began with our support and also support of Bristol Myers Squibb in March of this year. And I'm saying that was quite heroic, both from the patient and the physician point of view, because it occurred right in the middle of the COVID crisis in Maryland. Yet Mark was able to start the trial in which he's going to try this vaccine plus the 2 checkpoint drugs, the same ones that were used in the German patient, and asked 2 questions. Can patients develop immunity against the cancer? And does that give clinical benefit? So we're following that with great excitement and sort of bated breath to see what happens. So let me just summarize what I've been saying. Is there hope on the horizon? I'm convinced, yes, there is. I think there's some rational approaches in targeted therapy. The idea of the chimera protein as a drug target, these signaling islands, or finding genes that the cancer depends on, or just a marker that we can use as a way to bring a smart bomb into play to kill the cancer. On the immune therapy side, the combination of understanding how and when to use checkpoint drugs in combinations that may be more effective in this cancer, targeting the chimera for its attack, finding other possible natural targets for fibrolamellar for the immune system, or using T cells as the payload in smart bombs. And there's a kind of therapy called CAR T where you could do that. So I think there are a whole range of possibilities. My goal as scientific director is really to just take a lot of rational shots on goal against this cancer with the belief that the technology is now moving to the point where a lot of that can be done. In that process, we have a marvelous network of scientists and physicians dedicated to work on this cancer. And we've formally in the last year actually asked 6 of them, partly through the CZI initiative, to join our scientific advisory board. First time the foundation's actually formalized that. Their names are here. They include some of the leaders in oncology, immunotherapy, targeted therapy, and cancer genetics. And they're the people who keep me informed of what we should do and really doing it all for the benefit of the heroes who are you, the patients and their families, who, with your courage, make all of this research possible.